Today, we'll be talking about automated familial search using a probabilistic genotype database. This is joint work between the Cybergenetics Corporation and the Kern Regional Crime Laboratory in Bakersfield, California. Let's define familial searching. Suppose a person, indicated by the blue square outlined in black, left their DNA in a crime scene with 3 other people. Who is he? We could find out by comparing his genotype against the database shown on the right. Is he in the DNA database? No, he's not. Shown on the right is the family of the blue square individual who left his DNA at the crime scene. No, he's not in the DNA database, but as shown on the far left with a dotted line around the blue square, his brother is in the DNA database. Comparing the evidence genotype on the left with the brother of the person in the database on the right, we can use his brother to find the person who left the DNA using a DNA database. That's familial searching, finding people who left their DNA in evidence through their relatives on a DNA database. The TrueAllele probabilistic genotype database is far more informative than old allele-based databases like CODIS. The newer approach uses all of the peak-height data, models variation in the PCR experiment. It can consider all genotype possibilities and model PCR uncertainty using genotype probability instead of partial allele profiles. Genotype match information is preserved using likelihood ratios instead of weak allele inclusion methods. TrueAllele database automation starts with a DNA database of reference genotypes. These genotypes are uploaded to a TrueAllele reference database. Then, the probabilistic genotypes of family members are added to the reference DNA database. TrueAllele infers and uploads these family genotypes to the database as shown in the figure. From the original person in the blue square, the TrueAllele infers and uploads the parent and child probabilistic genotypes, indicated PCH as the abbreviation, as well as the sibling genotypes, indicated SIB. Suppose a person leaves their DNA at a crime scene, as shown in the 4-person mixture DNA evidence on the left. Now comparison can be made with a DNA database that's enhanced with the family members represented as probabilistic genotypes that are also available for comparison on the DNA database. Now the person in the evidence can be found through their relative's DNA on the family-enhanced DNA database. The database matches the separated probabilistic genotype from the DNA mixture evidence with a probabilistic genotype from a family member on the enhanced DNA database, producing a positive likelihood ratio association. A relative of the crime scene person has now been found.
Kern wanted to validate the TrueAllele software kinship application for forensic casework. Toward this end, Kern conducted a series of validation studies measuring accuracy, sensitivity and specificity, and known and non-probative evidence. In the accuracy study, Kern sought to demonstrate how the kinship application could match various inferred kinship genotypes to single-source evidence genotypes. To do this, they had DNA samples from 4 families, as shown in the figure below. On the left, family 1 has 12 known individuals spanning 3 generations. Next, family 2 contained 4 known individuals, all immediate family members. Then, family 3 had 6 known individuals, with half-siblings included. Finally, family 4 had 5 known individuals, all of whom were immediate family members. Looking at the results of the accuracy study, these 26 samples provided 676 pairwise comparisons for 3 different assumed genotype groups. The first group, PCH, involved inferred genotypes of parent or child of the known. The second group, SIB, included inferred genotypes of full siblings of the known. The third group, HAS, included genotypes of half-siblings, grandparents, grandchildren, aunt and uncle, and niece and nephew of the known. In total, these 3 groups provided more than 2,000 comparisons. Looking at the results, it was concluded that evidence to parent-child and evidence to sibling searches could possibly be used to identify matches between single-source evidence items and inferred parent-child or sibling genotypes of the parents, children, and full siblings of the true contributors. In the sensitivity and specificity study, TrueAllele inferred genotypes from parent, children, and siblings of true contributors were compared to various mixed DNA samples to evaluate the rate of true matches and the corresponding match scores. 31 single-source profiles and 36 mixed samples ranging from 2 to 5 contributors were compared to the inferred genotypes of 44 single-source known reference profiles, including many relatives of true contributors. Looking at the sensitivity and specificity of evidence versus parent-child searches, those searches appeared to be more specific for inferred genotypes of closely related relatives of known contributors, as shown in the table below. Also, looking at the sensitivity and specificity of evidence versus sibling searches, those searches were more sensitive for inferred genotypes of more distant relatives of known contributors, as shown in the table.
A third component of the validation was a non-probative evidence and known study. This used a 2012 sexual assault cold case. There are 20 DNA evidence items, including left and right bra cups and an unworn shirt with a non-sperm fraction and a sperm fraction. There were also 2 non-suspect known references: a consenting partner, CP, and the consenting partner's father, CPF. Routine TrueAllele casework DNA analysis found matches between the evidence items and the references. The left and right bra cups both matched the consenting partner's DNA profile, and the sperm and non-sperm fractions from the shirt matched the consenting partner's father. There were also another 16 case items, none of which matched either the consenting partner or the consenting partner's father. The Kern County Laboratory conducted an automated familial search using their family-enhanced TrueAllele DNA database. Shown here are DNA match results with likelihood ratios over 1 million. The enhancement was a reference that had the consenting partner's inferred parent-child probabilistic genotype. The question was whether the evidence would find the father of the known consenting partner. Listed on the left in red are 4 evidence items, 2 from the bra and 2 from the shirt. All 4 of these were matched by the automated TrueAllele familial search database to the consenting partner's inferred father genotype. This demonstrates the sensitivity of the automated familial search mechanism. Moreover, another 5,000 DNA database evidence items did not match either the consenting partner or his father. This shows the specificity of the automated familial search database approach. We have shown the vision of a fully automated familial database, of the kind that Cybergenetics implemented for the World Trade Center disaster 15 years ago to identify human remains. The idea is to infer mixture and kinship genotypes up to probability and compare them. It's a fully automated process there need be no human involvement. On the left, we see how TrueAllele can separate complex mixtures into the probabilistic genotypes of each component contributor. That's from a crime scene. On the right, we see how reference samples on a DNA databases can be expanded into the family genotypes of their unknown relatives. And then comparison can be made automatically between the evidence genotypes and the family genotypes in order to determine database matches producing statistical likelihood ratio numbers in order to find the relatives of people who've left their DNA at crime scenes in the TrueAllele database. In conclusion, familial search finds investigative leads. This talk discussed automated TrueAllele familial searching on a probabilistic genotyping database. The Kern Laboratory developed protocols for this approach and tested them on 3 familial search validation studies. They found that the parent-child inferred genotypes were more specific than the sibling inferred genotypes. The approach is very cost-effective compared to current methods because it reuses existing DNA evidence data. It's also much less labor-intensive because using automation, the familial database search becomes routine. We have introduced a computer-automated familial search workflow. Thank you very much.